right, so this is, we're continuing. I guess we may merge these videos, I'm not sure, but if not, this is video 2 of muscle tissue from chapter 10. So here we have, what I've drawn in is kind of a composite picture up here. So what you see is this line right here represents the sarcolemma. We already dealt with that. This would be a T-tubule, but as you can see, I've got a bunch of text I need to write. This is the axon terminal, neuromuscular junction, etc. We've been here. Now, we're just going to start off. The muscle's relaxed, so I'm not contracting it, it's relaxed. And at rest, and that'll be a good thing to say, at rest, you're going to find a lot of sodium outside of the muscle cell and a lot of potassium on the inside. You're also going to find a lot of other ions, including a lot of anions or negative ions that are inside of the muscle. So the net electrical gradient that you have at rest is that the inside is got a negative charge compared to the outside. So the resting potential, and that's a term, that term potential we always use as something else, right? Like that kid's got a lot of potential. But the potential in this case is just a charge differential. So there is a resting potential, there's a resting charge differential from one side of the membrane to the other that is negative on the inside. So at rest, the membrane potential is about, and here's the graph I've drawn in, is about negative 90 millivolts. So the membrane potentials that charge across the membrane. You don't have to remember these numbers. I'm not going to ask you about the numbers because they vary from one muscle type to the next and from nerves to muscles. So just know that it's a very strong negative, which means that there's a lot of negative things, a lot of on the inside, a lot of positive things on the outside. Even though you see positive things on the inside, the negatives dominate on the inside. So from the inside to the outside, you've got a negative charge on the inside, a positive charge on the outside. When I secrete the neurotransmitter, opening up these ion channels, well, the first thing that's going to happen is it's going to open up the sodium channels. So sodium's going to rush in. And as sodium rushes in, I'm just going to kind of clutter my picture here. As sodium rushes in, you get more positive ions on the inside, which means that that membrane potential is going to tick up like this. So you're going to have the membrane potential becoming less negative at first, I should say, and then it crosses into positive territory and reaches this point, which we've ballpark would be about plus 35 millivolts. So the outside now the inside I should say, is positive compared to the outside. We've accumulated a lot of these positive ions on the inside becomes positive, the membrane potential shifts to a positive, and this is what we call the action potential. So here at negative 90, we call that the resting potential. And then here up at positive 35 or so, we call that the action potential. So the action potential is a charge just like the resting potential. Now this process of going from the resting potential to the action potential is called depolarization. So the membrane depolarizes when sodium channels open up and sodium rushes in. I'm going to give you guys the overview image here on the projector in a minute. We've reached the action potential, and at this point, I'm going to open up potassium channels. So now, potassium is going to start leaking out into the extracellular space. Well, if all the positives are leaving from inside the cell, the inside of the cell is going to become more negative. So as the positive things leave, it becomes more negative, and they call this process, as this voltage electrical gradient returns into the negatives, they call this repolarization. And that membrane potential is going to go all the way out down here till it actually bottoms out below the resting potential. This state right here, which we don't have a value for, this state of the membrane is called hyperpolarization. So it's hyperpolarized at this point. And at this point, you're going to have something that's going to get it, you're going to have some an active transport pump return it back. Let's see if I can draw this. So it's going to return it back to this resting potential, and I'll talk about that in a second. And at this point, once you get back to your resting potential, you can do it again, right? Now, once you get back there, your sodium's back on the outside, your potassium's back on the inside, and you can do that same thing again. But let's look over here at this image that I put up earlier. Now, hopefully this will make more sense at this point. So what we have is our resting, RMP, resting membrane potential. Resting potential is fine. And you see our axes here. Y-axis is millivolts. X-axis is time. And the time across which this occurs is really brief. These are milliseconds. So this is 3/1000ths of a second that your muscle's going to have this electrical phenomenon take place. So sodium channels open up. Sodium channels open up. Membrane depolarizes, and you reach this thing called the action potential. At that point, potassium channels open up. Potassium then bails out of the cell. Repolarization occurs, and you get the membrane potential returning back to the negative here, where it's called hyperpolarized. Now that hyperpolarized state doesn't last forever, as you can see on here, and this is going to be the job of something we call the sodium-potassium pump. Now, I'm going to fill it in over here. I'm just going to kind of draw it. It's not that great, but let's just say that this has occurred. Sodium has rushed in, and potassium has rushed out. So there's a simplified view of it. Depolarization has occurred, and repolarization has occurred. Well, based on the graph, that's where we're at right here, right? Our membrane potential is way down here in the negatives again. But the problem is that our actors are not in their correct places, right? We started off with sodium on the outside, potassium on the inside, and after all this, we end up with sodium on the outside. Sodium on the inside and potassium on the outside. So we've got our players in the wrong spots, which is the job of this active transport mechanism called the sodium-potassium pump. And I'm just going to draw this kind of blob in here. It's a terrible rendition, but whatever. Well, this pump's job is to get these guys back outside and to get these potassiums back inside. Well, it kind of does that, and you can see graphics. If you want to look up sodium-potassium pump and look for videos of it, you can see it in motion. But suffice to say that it's going to move the potassiums back inside, and the sodium's back. It's going to move the sodium's back outside and the potassium's back inside at a cost. Okay? Anytime you're pushing things against their concentration gradient, and as you start stuffing those sodiums back outside the membrane, you're pushing against the concentration gradient. And as you start pushing the potassiums back inside, you're pushing against the concentration gradient. It's going to cost ATP. So this is one of many taxations on your ATP, on your energy supply, that you have to deal with every day on a constant basis. A huge percentage, actually, of your daily calories are spent on just putting sodiums back on the other side of the membrane and potassiums back on their side. Add that to the actual calorie cost of moving a weight, right? Of moving the muscles, and you've got a significant output of calories. So let's look at that. Moving here, there's a couple of things when you're talking about muscles that require ATP. First, we talked about in the last half of this video was the cross-bridge cycling. Now that's a term I didn't use before, but cross-bridge cycling is just, if you remember my hand motions here, cross-bridge formation, power stroke, detachment, resetting that myosin head, cross-bridge formation. This is not free. This process is not free. It costs ATP. And cross-bridge cycling is just basically the myosin walking up that actin filament, making your muscles contract. Costs energy. And then the sodium-potassium pump, which I just described. So when it comes to your muscles, you have two main costs. Well, how do we get this molecule back? We spend ATP. If you remember from our metabolism section when we were doing in-person lectures, talked about the glycolysis and the transition reaction and Krebs cycle and electron transport chain. Well, those are all steps in how you make ATP, and ATP is the molecule you're going to spend here to do this, and you're going to spend it during the cross-bridge cycle. So how do we get it back, or where do we get it from? There's three basic ways or methods. One, you just store it. So if you're at rest, if you're just sitting around or sleeping or whatever, watch TV, you're not contracting a lot of skeletal muscles to any great intensity. So you're going to build up your ATP store. So you're going to kind of get some ATP stored in the muscle, get it ready to contract if you need to. And there's also another molecule here called creatine phosphate. You may have seen creatine in like supplements for weightlifting. Well, creatine phosphate's a molecule that you store in your cells, which has a phosphate group. And if you remember how ATP works, is that you've got an adenosine nucleotide here, which I'm not going to draw, and it's got some phosphate groups attached to it. So this is ATP right there. When I break that third covalent bond there, that 1, 2, 3, yeah, covalent bond between the second and third phosphate, this is going to release a lot of energy, right? So, which I can then use to contract a muscle or, do the sodium potassium pump, etc. But I need to get that back. So whenever you see that formula that goes ADP can go to ATP. Wait, I should got it backwards. Here's what happens when you're trying to think off the cuff. ATP can be broken down into ADP plus a phosphate. Well, that's that guy there. So if I'm going to reattach it, I'm going to get it from a number of different sources. One of them is this molecule, creatine phosphate, which happens to have a phosphate group ready at hand. So if creatine phosphate finds an ADP, one that's missing a phosphate, it can go, here, buddy, and give him his phosphate group. And so that's a kind of a real rapid way to regenerate or use ATP. And this will last you about 5 to 6 seconds. You have about 5 to 6 seconds worth of ATP or creatine phosphate in your muscles ready to go. Well, that's not a long time. So if you're starting, let's just say you're doing a sprint or you're running some stairs or you're doing some deadlift. Well, you're going to be able to crank out some ATPs anaerobically. And we mentioned this in the previous section, weeks ago, where you have the capacity to just chop those glucoses in half. So if I take a glucose, chop it in half, form it into 2 pyruvates, I can get a real small dose of ATP out of that. I can get about 2 ATP per glucose. Well, 2 ATP per glucose is not that efficient. It's only about 5% as efficient as aerobic respiration. But it's about 2.5 times faster. I can crank out that ATP really quick. So it's like you're kind of, got your carburetors wide open and you're just blowing gas through there. You're costing yourself a lot of energy in the fact that you're wasting a lot of glucose or spending it inefficiently, but you are getting a lot of power out of that, which is why when you lift that dead big heavy weight, or you take a sprint, or you run some stairs, or you're doing something hard, you can get a big burst of energy out of yourself for about a minute, right? You can go about a minute on anaerobic capacity. If you've ever sat there holding up something heavy, let's say you can do, hang from the monkey bars the longest, eventually your hands are like, and they'll give up, right? Because you've basically exhausted your anaerobic capacity. Now, aerobic metabolism is what we went through with all of those steps, the glycolysis and the Krebs cycle and so on. So aerobic metabolism you can do basically indefinitely. So this whole time that I've been lecturing, I've been conducting aerobic metabolism. I'm not doing a whole lot of aerobic exercise per se. I'm not tiring myself out or anything, but I am expending energy at a very metered rate. I'm not going to run out of gas after a minute and be like, I gotta take a breather, right? So very efficient. You get about, if you remember, you get about 38 ATP per glucose. That's optimal, but 38 ATP per glucose is a lot more than 2. And but it is a little bit slower, which is why if you have to do a high intensity exercise, you start tapping that anaerobic capacity significantly. Remember that full glucose metabolism occurs where you take glucose, break it into pyruvates, send it into the mitochondria, do all that. You're not gonna be tested on that, but that's what I'm talking about. And then lastly, that's why they call it aerobic, is because you need that oxygen as the final electron acceptor in electron transport. And that's why we need to breathe oxygen. A big picture view there. All right, let's move on to the concept of oxygen debt. If you've ever done some high intensity exercise, if you've ever, when you were in grade school and they forced you to run a sprint, or they forced you to do some rope climb or something like that. Well, if you've ever like gone up a set of stairs and you get to the top of the stairs, let's say you really pushed yourself and you stop climbing or you stop sprinting or you get done with that set of deadlift and you're going like this, right? You're not doing anything anymore. You're not still deadlifting. You're just gassed and breathing heavily. Why would you want to breathe heavily after you're done exercising? Well, it's because you went into something called oxygen debt. In oxygen debt, you're going to have to repay. You're going to have to pay the piper. Okay? You're going to have to convert the lactate that, if you remember, was a breakdown product of pyruvate, and that's what you basically get rid of when you're doing heavy lifting. You're going to want to reconvert that lactate to pyruvate so you can use it in the normal aerobic respiration process. That takes ATP. That takes aerobic respiration. You're going to want to store up any glycogen that you depleted when you were exercising. So storing glucose as glycogen in your muscles and your liver costs energy, which means that you're going to have to breathe. And then restoring these creatine phosphate and ATP stores also requires it. So you're going to spend a minute or two huffing and puffing because you're going into straight aerobic respiration at that point. So even though you were sprinting and probably huffing and puffing while you were sprinting, you're still going to have to huff and puff. Huff and puff. Isn't that a house in like Harry Potter? Hufflepuff? You're hufflepuffing for quite some time afterwards because you're tired, right? But then after you recover, you breathe normally. You may notice that when you're in good shape, you don't go into as much oxygen debt. If you've ever seen one of these people run a mile, or they run a 10,000, or they run a marathon, and they're just going, they're hell bent for leather towards the end or during the whole race, right? But these people are in such good shape that they get done, they're like, finish line, and then they'll do an interview. They might do this for a sec, and then they'll go like, well, I had a good race, started off a little slow. Not me, right? Not most of us. Okay. Let me move to this little section right here, this little table I've made here. There are 3 basic muscle fiber types. And remember, fibers are cells. 3 basic fiber types. Classes. There's slow oxidative, and fast glycolytic. Well, these terms describe themselves. Slow oxidative, that's going to be a slower twitch fiber that basically requires oxygen or aerobic respiration. Fast oxidative, it's a bit faster, still heavily reliant on aerobic respiration, therefore the name oxidative. And then fast glycolytic, fast fibers, fast twitch fibers that glycolysis, break sugars. So these guys are going to be specialists in anaerobic respiration, and I've kind of summarized their abilities here. And they really organize nicely. The slow oxidative have really high endurance. These are the ones that I'm using right now when I'm talking. These are going to be the ones that if I go for a walk or even a slow jog, they'll be functioning for the majority of that time. And I can jog slowly for 5 miles and not get gassed. Now, if I go a little faster, I'm going to have to recruit some other guys. But I can talk and walk and conduct normal daily activities for a long period of time. They don't. The next guys, fast oxidative, have a slightly less endurance, and then the fast glycolytic, these are the ones used during dead sprints and heavy weightlifting. Contraction speed, let me switch sides. So contraction speed, these guys are slow. This is why really super long distance runners oftentimes don't have a great sprinting ability. Now, if you're an Olympic 10K runner, your sprint is going to be better than most people's just because you're a super athlete. But you're not going to be able to beat the dead sprinters because you've got a large percentage of these slow oxidative fibers. Fast oxidative somewhere in the middle, and fast glycolytic are very fast contraction speeds. So this is what you're going to use to jump or sprint or when you're snatching a weight off the ground. And then for aerobic capacity, high, medium, poor, kind of works out logically. The slow oxidative use a lot of aerobic metabolism, fast oxidative somewhat in the middle, and fast glycolytic poor, right? So these are the ones, this is why when you do 10 deadlifts with a really heavy weight, you're cashed after that, right? That's it. you're going to take a couple of minutes off and let those guys recover because they don't have the ability to burn oxygen slowly and at a pace. All right, switching topics again. Now I'm kind of jumping from topic to topic here, and I'm going to jump up to this little table up here, which talks about muscle tone. We've all heard about muscle tone. Say, well, what is it? A lot of times when I ask students in person, they'll say, if I say, who wants to take a guess? They'll raise their hand and say, how firm your muscles are. Yeah, okay. That's a result of muscle tone. So the greater muscle tone you have, the more firm those muscles feel. They don't feel as saggy, flappy, sloppy. What's the word I'm looking for there? But what it is that at rest, like if you relax your muscles, a small percentage, let's say a muscle's got 1,000 fibers, that's a small muscle. But if they had 1,000 fibers, there's going to be 5 or 10 or 15 or 20 of those fibers, a small percentage of them that are going to be contracted at any given time. Even though my biceps muscle is at rest, some of those fibers in there are contracting. Now, the more you exercise, the more fibers contract at rest, which is why people that are, in good shape or whatever, or fit, seem to have more firm muscles because they have greater muscle tone, which is the percentage that's contracted. Why do you want to contract muscles at rest? It prevents injury. The muscles are kept slightly warmer. They're ready to go a little bit better. So people that are regularly active, their bodies kind of interpret that as, I better be ready because this person's regularly active. If you're not active, if your biggest exertion is reaching for the remote as far as you can, then you're probably not going to have as high of muscle tone, which means your muscles are going to be a little bit softer. Now, this contraction is not free. So having a greater muscle tone means that you're burning more calories at rest. So if you think about it in the big picture, if you exercise or if you run and work out or whatever, that's going to make you burn more calories at rest. So people that are in better shape consume more caloric energy over a given period of time, even when they're sleeping or watching TV or whatever, playing a board game, than someone who doesn't work out. So good tip for you if you're trying to lose weight is to work out. That's pretty obvious, right? That's not an insider tip. Okay, moving over. Here's some interesting terms that we'll finish off this chapter with. Ooh, I forgot. Remind me to talk about rigor mortis. Not the camera, but the person behind the camera. Isometric versus isotonic contraction. So isometric contraction, and you may have heard these as isometrics, is where you'll do one of those. Can you still see me in my film? Where you do that, like that wall sit thing, right? You've ever done one of these? I'm not moving. My arms and legs aren't moving anywhere. They're kind of stationary. But I can assure you that by about now, I'm starting to realize that my quadriceps and my glutes are contracting, right? So this stationary muscle contraction costs ATP. This is those cross bridges. Those cross bridges are locked in like this, and they're not crawling up or relaxing, but they're stuck like that. Okay? This process right here requires electrical impulses from my neurons, requires action potentials on the sarcolemma, and it costs ATP. It's also going to break down sugars, basically do a lot of anaerobic respiration. So stationary contractions are called isometric. They still cost energy, and they still work out your muscles. So if you ever want to work out, if you're ever lifting weights and you want to test that out, like take that bar, and when you're doing a bench, like try and leave it there for a minute. See how you feel, right? And by the time you give up, you're probably going to hope you have a spotter because you're not going to be able to put it back up. Which brings me to the next point. Isotonic contractions are moving. So these are going to be change in muscle length. And your classic one is concentric. So if I'm going to do a barbell curl, I'm going to shorten the biceps muscles. And so that's a concentric contraction. Concentric is a subset of isotonic because it is changing length. Concentric contraction is shortening. Now, I told you right at the outset that muscles don't forcibly extend. But here is eccentric contraction, which is lengthening of the muscle. The muscle's not, so my biceps brachii muscle is contracted right now. Well, if I put a heavy enough weight on there, it's going to pull my arm down, right? And it's going to stretch, that muscle's going to be getting longer, but it's contracting the whole time. It's trying to stop that. So you can kind of think of it like like a cat trying to climb up a tin roof, right? It's like losing ground because it's sliding. So the weight's so heavy that those myosin heads are not able to do the job. Those eccentric contractions, this is actually eccentric contraction right there. As long as I resist that thing, it's eccentric contraction. Eccentric contractions are about 50% stronger than concentric contractions. So if you've ever been in the gym and had a spotter, let's use bench press as an example. So you'll go 2, 3. So you're getting these weights done, you're getting these reps done, and you're getting hard. So it's like 8, right? And they'll go, come on, one more, or do a negative or something, right? So that's when you go down real slow and you're trying to control that weight's descent. That is an eccentric contraction. And if you're gassed, when you get to your chest, you're not going to be able to put that thing back up. So these guys are strong enough to control that weight when it's on its way down, but the concentric contraction wouldn't be strong enough to put it back up. So that's why you need the spotter, and then if they're a real jerk, they'll make you do it one more time. All right, here goes the second to last thing besides rigor mortis, effects of exercise and aging. They're not really connected, but we got them in the table, on muscle. So aerobic exercise, so aerobic cardiovascular workouts, go for a run, get on the treadmill, go swimming, whatever, hiking, if it's a vigorous hike, you're going to get some aerobic exercise. Here's a big word for you, angiogenesis. Well, genesis, we know that's the beginning or making or something like that. Angio refers to blood vessels. I won't go there. So angiogenesis is making blood vessels. You actually, when you exercise aerobically, you make more capillary beds and you get a better blood perfusion of blood to the muscles that are doing the exercise. This is why when you decide to start a regimen of jogging, the first day or 2 or 3 or 5 or whatever is really difficult. Your muscles fatigue really badly, right? In the middle of that jog or run, you'll feel like muscle deadness because they're basically running an oxygen deficit. But if you're getting in good shape, that same exact speed is easy for you like 3 weeks later or 2 weeks later. It becomes much more easy because you've built up a better supply chain to those muscles. You also increase the number of mitochondria. And as we know, mitochondria are the aerobic respiration sites inside of your cells. So you build more blood vessels to the area, bringing more oxygen, and you build more places to use the oxygen. So it's really amazing how much better you can get it at exercise by exercising. This is most dramatic in oxidative fibers. So the oxidative muscle cells that use oxygen show that result the most. And you can even get conversion of fast glycolytic to fast oxidative cells. So if you do long and slow exercises, if you do long runs regularly, and you're not really sprinting any time, you're going to get a lot of these oxidative fibers. Now, distance runners, if they're not really fast, if they don't have a kick, is what they say, will have to speed train as they run up to these events. So as they get closer to events, they'll start working hundreds or quarters or whatever, where they'll try to convert some of those guys back into fast glycolytics, so in case they're in a sprint at the end of the race, they might have a shot. Anaerobic or resistance exercises, basically example weightlifting. This is where you get hypertrophy, where you get bigger. Hypertrophy is just bigger muscles, which are due to bigger muscle cells. You don't actually make more muscle cells. Now there's a thing called hyperplasia, I won't test you on that, where the muscle cells can split, so effectively kind of double or increase numbers, but you're not getting mitosis, you're not getting division of the muscle cells to make more cells. But you do get, the muscle cells get fatter. So if a million muscle cells all get a little bit fatter, the whole muscle gets fatter. And that is due to an increase in the number of actin and myosin filaments, those filaments inside there. You get more of those, bigger cell, bigger muscle, stronger, etc. Now if these guys are all, if you do nothing but like heavy lifts and stuff, you're going to get strong, but you're not going to increase your endurance that way. If you don't use it, you're going to lose it, right? So atrophy. Atrophy is this basically when the muscles get smaller. If you worked out like crazy for 6 months and you're all yoked and stuff and you're checking yourself out in the mirror and you're like, yeah, what's up? And you're doing that stuff, you know, we all do, maybe not. But if you're doing that and then you're like, woo, and then something happens in your life where you can't work out for a couple months, you're going to go, you know, you're going to shrink back down. Contrary to popular belief, muscle doesn't turn into fat. It's not going to just bloom, turn into fat. It doesn't just convert. Now, if you're eating the same amount of food and you're not working out, that energy is going to go towards making fat cells rather than muscle fibers. But there's no direct conversion of one to the other. You're going to lose muscle tone, strength, and size. So at least do some maintenance. And then aging, as you can see, someone's written here, sucks because the results kind of decrease with age. It starts in your mid-30s, and it doesn't get any better. But you can, there's no such thing as give up if you don't give up. So you can keep working out, you can keep running, and you're going to be able to maintain that a lot longer than someone that's not working out and running. Same thing works with atrophy. If you take a year off, you can get back in it. You just gotta convince yourself. Okay, that's the end of this, but I do want to talk about rigor mortis, which is something I wanted to mention much earlier. So picture this. I'm going to get down here, and here's our, I'm going to draw a little quick and dirty cell with the, myofibril with the sarcoplasmic reticulum here, right? And I said that it's got calcium. Well, the calcium leaks out. Calcium leaks out and goes and gets on, activates troponin, which moves tropomyosin, which opens up the binding sites, which lets myosin attach, right? So as long as calcium's out here in the sarcoplasm, these guys can do this. Am I on screen? All right. So these guys can do this. Now, let's just say I'm going to send an impulse to my biceps to contract. Hiya! They did it. Okay. Now I'm going to stop that electrical impulse. Stop the electrical impulse. Think about what's happening. The sarcoplasmic reticulum's not being stimulated. There's no action potential. There's no opening of these calcium ion channels. So the calcium all gets pumped back in, and that's something I didn't mention before. But when you're done sending that stimulus, these doors shut, and the calcium all gets shoved back inside. So that calcium is going to be shoved back inside. But that requires ATP. Are you making much ATP when you're dead? No. So if you die, your ATP production machinery stops. And in order to pump calcium back from out here into the sarcoplasmic reticulum, you need ATP. So when you're dead, calcium accumulates out here in the sarcoplasm, which activates troponin, which moves tropomyosin, which allows the cross bridges to form. And these cross bridge doesn't require ATP to form. That's the little subtlety that I was talking about earlier. This is free. This costs energy. So if this is free, when you're dead, those myosin and actin filaments get stuck together. So when somebody dies, after a short period of time, they exhibit what's called rigor mortis. And that's rigor is stiffness and mortis is dead, so dead, stiffness of the dead. Where those muscles will kind of contract. Generally the flexors kind of win a little bit. So you'll get somebody dead and they'll kind of be kind of stiff like this, right? Depending on which position they kind of died in or were forced in. So that'll remain for a couple of hours, and then after a few hours it'll start to loosen up, not because of the ATP going away, but because that the proteins start to degrade, you start to decompose. So after several hours, those muscles loosen because they're becoming they're not rotting yet, but the proteins are degrading. It takes constant effort, constant energy just to keep all this stuff working, right? So you need to constantly eat food, constantly generate ATP, and that's the sort of sequence of events after you die. All right, that's it for. I'm skipping smooth muscle and cardiac muscle. Yeah. Shoot, how did I skip that whole section? I'm not done yet. Let's go over here. Man, how did I almost forget that? It's because I didn't write it up there, did I? I did not. All right, let's. I'm going to turn off the lights just so we can see this clearly. Ooh, that's super clear. Is that okay? Is that good? All right. So now, sorry, it would have been better to do this earlier, but it'll work. So here's our action potential diagram. Now I want you to contrast this diagram. It's real angular, so it looks different, but you'll sometimes see it. When I drew it a little bit more gradual with this diagram. Now they look very different, but the diagram that I drew of the action potential looked similar. Notice the axes. This is an actual muscle twitch. This is a measurement of tension on a muscle that a muscle fiber exerts. So here's tension on the y-axis compared to voltage on the y-axis. This is a graph representing how strong your muscle is pulling over time. This is called a muscle twitch. When there is a stimulus, when that neurotransmitter gets across that neuromuscular junction, there's going to be a slight delay here, and you see this in dark blue, and that's called the latent period. And that's a little lag time between stimulus and contraction. Then you undergo this period of contraction where the muscle shortens, and that's myosin grabbing to actin and crawling up it. Then when you cut off that stimulus, you're going to get a period of relaxation where the muscle returns to its normal rest and resting length. So this is a muscle twitch. Lag period, latent period, period of contraction, period of relaxation. The whole thing's called a myogram, a graph of a muscle. Now I want to look at some muscles and their twitches. These are twitches of 3 separate muscles. This is the lateral rectus muscle, which you haven't gotten to yet. This is an eyeball muscle. This is the muscle that makes your eyeballs go left and right, depending on the eyeball. But if you've ever looked around at something or watched somebody's eyes move, those eyes jerk from place to place, right? They're really quick. These are a very fast twitch, fast glycolytic muscle. Now, a muscle that has this is a big muscle right here, but it's got a lot of fast glycolytic, but it's not almost entirely fast glycolytic. This would be your gastrocnemius. And your gastrocnemius is one of your calf muscles. This is the outer calf muscle. This is what you think of if you think of your calf muscle. So it has a pretty quick response time. Still has a latent period. It's in here. Period of contraction, period of relaxation. But it's slower with the calf muscle. This would be the muscle you use when you jog, but you really use it when you sprint. Now the soleus is a calf muscle that lies underneath the gastrocnemius and is a much more slow oxidative muscle. This is the one that you're going to rely on heavily when you walk, or perhaps when you do a slow jog, but not when you do, box jumps or long jump or sprints. This would be the sprinter muscle. This is sort of the jogger or walker muscle. You can see that the period of contraction is much longer, and it's a lot, longer time to get through a single twitch. This is why you get, fast twitch versus slow twitch. And lastly, for real this time, here's a twitch. Okay, so this is just basically a small version of this. There's the twitch right there. Here's what happens if you twitch and relax and twitch again. And twitch again maybe. What you're getting is this process called wave summation. And I don't want you to worry too much about the difference between wave summation and unfused tetany. Big deal. It's just that if you twitch halfway through the relaxation, your next twitch is going to be a little bit stronger. Reason being, the muscle's already partially contracted. It's already warming up. you got increased blood flow to it, so it's going to get stronger with these successive twitches. Now, what you want to notice here is this is the frequency between the stimuli. As frequency increases, this doesn't really show that, but as you send these stimuli down there to that muscle in succession, you're going to get stronger and stronger and stronger and stronger twitches. They call this incomplete tetany or unfused tetany. You may be familiar with the term tetany when it comes to tetanus or getting a tetanus shot. Tetanus is like locked muscle, but as you build up to that complete tetany, you're maintaining contraction strength. You see still that you've got some tension. The tension never completely releases, never go back to zero. Tension never completely releases, it just slowly builds until if you make those stimuli go very rapidly, so I'm going to try to hold up a heavy weight. I'm going to send many stimuli, which will then basically allow that muscle to remain constantly contracted. Now, eventually you're either going to stop the stimulus and the muscle will relax, or you'll experience muscle fatigue due to any number of reasons which I won't go into, and the muscle will fail. And then you'll go into oxygen dead, I bet. Okay, so know those terms. Twitch, let's not really discriminate between wave summation and unfused tetany, unfused or incomplete tetany. Let's just say that both of them rely on increased frequency to build stronger contractions with minimum frequency, the most rapid signals possible will allow the muscle to remain continuously contracted. Let me go back over here, turn on lights. All right, let's go back to me for a second. So that's chapter 10, and I'm just going to call it quits there. The next test will consist of joints, which I had already covered, this one, which is chapter 10, muscles, and then I'm going to do a little bit on chapter 11. I'm going to make a short video over chapter 11 just to cover some loose ends. I'm not going to ask you to know a bunch of the major muscle groups because that's what lab's for. So this is lecture 2 of 3 for the third test, which will be, I'll publish that whenever we get done with the material. All right, thanks.